everyone, this is the second tutorial, an introduction to Arescon. I thought it would be a good idea to make a new video since there have been many updates since the first video. There are many new tabs that are quite useful actually if you're running, for instance, colocalization, or you want to map some ROIs to already designated regions using Fiji, for instance. But also for the people who haven't used Arescon yet but they're interested, I thought it would be a good idea to have a shorter version this time, a shorter tutorial. However, you can always refer to the first one, which is more detailed. I'll still try to explain all the basics and all the essential qualities that you need to know in order to understand what's the reason we're using Arescon and why is it useful. So in the previous video I've been using a small but whole in a sense dataset. In this one I will just be using a demo version. The difference between the demo and the whole dataset is that in the demo we'll just have one plane. Now normally you can use Arescon for one plane, but usually it becomes more robust if you use more planes, and I will explain why again, of course. However, because I don't want to waste time, I will be using one plane, but when I want to show you what's going on the larger scale, I will be shifting to the whole dataset, and from the whole dataset I will be showing you some things. I hope it's clear enough. So without further ado, let's first explain what is the reason that you should probably use Arescon. I'm going to open an image. And we're talking always about pretty much 2D images. This is a 3D image in a sense, because it's a stack, right? Let me make it more clear to you. So if I go adjust brightness, you see that we have the same part of tissue and its image across slightly different focal points. Now this doesn't really mean that we're having a 3D representation of a brain, as we do, for instance, when we're doing brain clearing and we go across different Z-planes, many different Z-planes. In this case, it's just a few Z-planes. And the reason we're doing it is because, as you can see, there are parts that you can have pretty well focused on one plane, but if you shift to another plane, you can see that the focus is lost, and you need to actually capture more points in order to make sure that you will have at least one crisp version of your cell's nuclei or whatever. So this is the first step to combat the problem of non-perfectly aligned on a particular surface objects, which is pretty much never the case, at least hasn't been for me so far. There are many factors that can lead to that. One is that probably the tissue becomes a bit wavy, although we don't see it by eye, this happens. Another one is that, of course, there are slight differences in terms of the depth of where an object lies. And the third one is that slices are not always taken sliced, completely horizontally. So it could be that there is an angle, and this will, of course, affect the way you image it. So, how can we get good ROIs for that region while at the same time we're getting good ROIs for that region, right? One way to go, of course, is starting with Z-max projections. And for those of you who have tried it, you know that you end up getting an image that looks like, let's see, it looks like that. So here I have another image. This is Cephus in this case. And what Z-max does, there are several ways to do this projection, but in this case, unless I'm horribly mistaken, it picks the brightest pixel across all the Z-planes for all the pixels in the image, and that way it recreates an image that has more accentuated, let's say, objects. And although it's really nice for visualization purposes, it hasn't worked for me nice when I was using cell pose, for instance, to detect cells. I can show you an example. So if I go, just a moment, if I go here and I find some inferences taken from cell pose, you can see that many of my objects are actually missing. And I suppose the reason that this is happening is that you have more artificial objects when you do the Z-max projection. It's a tough word to say. And this probably affects the inferences and the ability of the network to reliably detect cells. Of course, having said that, you can always try training your cell pose network with images that are Z-max projected already, and that might resolve the problem. However, because in my case I needed a lot of precision, I decided to go for another way, and I decided to go for Arescon. I just want to show you a few more problems with Z-max before I move on. So with Z-max, many times the contours of adjacent cells are a bit distorted, and this makes it harder, of course, for the network to detect the right contours. And if I manage to find a way to close this, nice. And here's another example whereby you can see that contours are very distorted when you have groups of neurons that are close by. Again, having said that, this doesn't mean that Z-max projections won't work, but if you need more precision, then you can use Arescon. Now, if you want to make your network more lenient to detecting more cells, you can do it from cell pose. There are variables like the cell threshold probe thing and flow something that I can't recall by heart right now. There are two arguments that you can use in the model eval that function in which you can get actually more cells. However, the problem with that particular option is that the contours of the predicted cells start to become less reliable the more loose your criteria of detecting cells become, and especially if you're looking for overlaps, this is going to make it a bit more tricky. Also, another problem is that you cannot reliably filter a cell based on the surrounding mean background of the cell, because the surrounding mean background is now also sort of artificial. It incorporates the brightest pixels of all planes nearby, and this is just not the best way to go, let's say. Max, so I can close these windows. And what we're doing in Arescon is actually we're collecting all these planes that you saw, right, in this stack of images, and we get inferences using cell pose across all planes. This way, we sort of ensure that we have at least one good capture, and most likely the most crisp capture of the neuron. The disadvantage, of course, is that if you move across planes, you will also get inferences for the same object when it is less clear, right? Because you have these different versions of the cell across the Z-planes. So what Arescon can do is that it can filter out the less clear versions and keep only one version for every object that lies across all these Z-planes. And another benefit, of course, with working with multiple inferences is that in a Z-max projected image, for whatever reason, you might get some false negatives. However, if you're running many planes, there should be at least one plane that will solve your observation. Even if, for whatever reason, an object has not been detected on a particular plane, it is very likely that it will be detected in a new one. All right. So, without further ado, let's open Arescon and let's see where you can find it. So, let's go to my GitHub page, and you can find Arescon on this link. Always recommended to check the releases and download the latest release. Right now it's version 5. You're going to need also Fiji for this, and the Fiji version you're going to need is actually part of the first version. So the first release has some test files and the Fiji included, as you can see. So from the assets of this one, you should download Fiji for Arescon, and this is a specific version of Fiji that you should use. Otherwise, the automated Pyoto GUI library that guides, let's say, um, the keystrokes and the clicks will not be able to work as it's supposed to do so. So please use this feature in particular. And the other program that you're going to need, and I will explain why when we got there, is the AutoHotkey. This again simulates some keystrokes, so you can go to download and you can download the version 2. Having said that, I think now we can start with Arescon. So you can find the executable version, and I strongly recommend to use this one actually, because it seems that it works as good as the Python version. So again, if you go to Arescon page and scroll down, you should be able to see this link that gets you to the executable version. If we click here, then you can go to show all files, and you can download the executable version and unzip it. It will take some time, but eventually you should have a file like this. So now if you go to the dist folder and you open the main.exe, it will take a few seconds, but Arescon should open after that. Cool. Now, I've inserted a few more options, so you can change the font size. It won't be perfect change, very smooth one. Different screens have different DPIs, and I'm not an expert when it comes to GUIs. So this is a way to make everything a bit more readable in case letters are quite extended. And if you don't like the graphics, you can turn them off. I will have them on for now. So as you can see, right, in ZStack images captured with slightly different focus, Arescon is able to isolate the most crisp version of each cell among all planes. Another very important function of Arescon, for the analysis that I'm doing at least, is that it can estimate the surrounding background of each region of interest, ignoring at the same time the signal originating from other regions of interest. And this is really important, especially when you're looking at nuclear signal, for instance. Many times it's hard to decide what should be filtered in and what should be filtered out. And this is much easier to do if you can get a very good estimation of the surrounding background. You can automate procedures and also make them less subjective, to be honest. Okay, based on the maximum planes that you have in your images, imagine that you have a data set with, I don't know, 10 Z-stacks, and one Z-stack has only 5 planes, another one has 10, another one has 15. So here you should pick the maximum. If you don't pick the maximum, then it will not process everything. In my case, as I told you, I'll just use one, and this is just for presentation purposes, right, for tutorial purposes. Strongly recommended to go for 16-bit. I've mostly tested Arescon with 16-bit, and unfortunately right now Arescon runs only on Windows because some paths need to change for the initiation of Fiji when it runs on Linux, and yeah, there might be some other issues on Linux as well. I hope I'll find some time in the future to also adjust this. Not now, though. Alright, so we start by selecting the folder with our images, and this would be a folder that contains all planes, right? So plane 1 images, plane 2 images, and so on. And within each subfolder, like plane 1 images, you have your IDs, correct? And these IDs are already split in planes. How do you split in planes? You can do it with Fiji manually. It's easy to find instructions. However, I also have some options here for you to make your life easier. So if you're taking, and you need to have both in that case, both multi-channel and multi-plane images, then by using this command you can initiate Fiji. Whenever you see this hand icon, it means don't touch anything, let Fiji do the job until it's done. And by clicking on that, it should actually split all your images to single plane and single channel images, right? And it should also give you the right structure in order to be able to continue with Arescon. If you also go here, you can see how this structure should look like. So you will be using a save folder, and based on the number of channels you have to have the respective number of CN files where n corresponds to number. So you have 3 channels, then you need to have a C1, C2. I see a mistake here. I should have written C3 in the third one, but you get the idea, right? So you have 3 subfolders, and within subfolder you have plane 1 images, plane 2 images, plane 3 images, and you need to have the same number of plane and images across all these channels. And in the end, what this function is going to do is it's going to split all your images and place them in there, which is the main input that you want when you want to continue here. Since we're in the Change Rows tab, I can mention here that all observations, all inferences that you get with CellPose are in a labeled mask format, so like this, not in ROI format that Fiji can use. And you can actually convert them to ROIs by clicking on this. There's a particular naming of ROIs based on which Arescon works. You can also find this on GitHub. By default, it will make, if you use the notebook that I sent you and this function, everything should go smoothly. So the notebook, in the meantime, you can find back to the Arescon page. I think I've actually posted it on top, yes. So strongly recommend, actually should use this particular notebook. This comes from the people of CellPose, of course, because Arescon works with inferences from CellPose. This one I also used a bit of AI to produce some code here, so it might not be the original file of the people, but it's certainly, like, all the information are taken by the AI from people using CellPose. What's important here is that there is this filter mask insertion to the names that I use to prevent getting names overridden in Google Colab in case there is a wrong option of directories. So for that, yeah, you just need to delete the filter mask from every image. This is actually easy to do. If you have Power Toys, I think it's an option of Power Toys. So let me see if I can find a folder and show you how it works. You can go to Power Rename, and then you can locate the part of the name that you want to delete, and it will do it for all files. So it's actually very easy to do. More information about this, you can go to the first video, but I won't spend more time here. Cool. All right. So let's go to the main inputs and let's add our images. We're going to the demo version. In the demo version, as I told you, I just have only one plane. So I'm selecting the images folder and I'm selecting the ROIs folder. And architecture should be the same for the ROIs. So these are all inferences taken from CellPose. And as I told you, recreate it if you also use that function. And what we're going to do now is that we're going to initiate Fiji, and Fiji will just be opening plane by plane all images. So if you had 100 images in plane 1, Fiji would open once. And if you also had 100 images in plane 2, Fiji would open twice. So this is not based on animal dependent, but it's just plane dependent, the times that Fiji will open and close. We have selected both. If you've done it correctly, you should also see that checkbox there. And next we're going to select the save folder, whereby our main measurements will be saved. I I can name it measurements maybe, or maybe main measurements to make it a bit more clear. Cool. Then you need to indicate the executable file of your image.j. Many times I say Fiji, but actually you need to use the image.j version. Otherwise it's not going to work. So this one, please. And the next one is quite important. This has to do with the delay that it takes, the time that it takes for your computer to open image.j. So as you can see, I'm clicking on image.j and it takes some time until it opens. Not many seconds, but sometimes it will take a bit longer. And what we're doing here is we're having a very custom-made pipeline that simulates keystrokes and clicks. Thereby, if you don't indicate a sufficient time, a sufficient delay, what's going to happen is image.j will open on the taskbar, because once you click here, your computer says that, okay, I'm running image.j, but your computer doesn't know when you've loaded image.j. And at some point, we'll have to click here and to start typing some commands. And unless you've given enough time for your image.j to open, then it will try to write the commands, although the Fiji will still be loading. And that's problematic. So please exaggerate here. In my case, I will set it to 14 seconds because I know 14 seconds is fine. But if you suspect that your Fiji takes, let's say, 10 seconds to open, maybe set it to 20 just to be on the safe side. And we're having a last look again at some warnings here. And when we ensure that everything is going to run okay, then we have to generate our measurements. Now, if I try to press this, I will get an error, and this is because you need to have image.j closed before you start, and any other files that you have potentially opened and have the name image.j on themselves. Okay, I'm closing, and now I'm going to run again. Now there is a fail-safe when you're using Pyoto GUI. Should work, I suppose, if you try to move your mouse on the top left of your screen. I'm not going to do it because I don't want to stop the auto GUI. But just to know, there is a failsafe, and I've encountered some bugs lately when the screen is locked. It seems that the failsafe seems to work. It's not really a bug. What I want to say by this is that please refrain from setting your computer to sleep modes, because if you do so, probably Arescon won't work properly. So you shouldn't have a sleep mode, and how you can adjust it actually is that if you press Windows and L, not L, sorry, I just locked my screen. I hope you can see my screen again. I think it's Windows and I, let's see if I'm correct, power and battery, and now we're going to this mode, and yeah, I recommend setting this to never, based on whether it's plugged or not, so that you ensure that your PC doesn't sleep. Cool. A bit verbal messages here. You will come across a few ones, but quite informative at the same time. So what it tells us is that we have files saved in the save folder. Let's have a look in the main measurements folder. So here you can see all the measurements that have been selected. And we have area, we have mean, and a few more values here. How do you set the values? This is image.j dependent, and you will only get the values that you've set measurements for. So you go to set measurements on image.j, and here you can select the values that you want. Now I'm using the basics. You should really have these ones, and you can add a few more if you want. Cool. Now that we've done that, we need to add the measurements to Arescon, because as you saw, we created an Excel file, but this Excel file is not read by Arescon at this point. So you need to add your measurements. Because I have just one plane, I'm clicking yes here, but this should be a warning for you if you're using more planes. So I'm clicking yes. And I think this is a good opportunity to mention that it's completely fine to run Arescon. Should have said that earlier, actually. It's completely fine to run Arescon with only one plane. If you haven't captured many planes to get a Z stack of images, and you believe that you have some decent signal in the region that you're investigating, you can still run Arescon. You just need to maintain the same architecture, namely plane 1 underscore images, plane 1 underscore rows. And also the names of your files. This is also, I hope I have mentioned it here actually. Yeah, you can see that all image names have to end to the respective plane number. This is something you can, again, you should have ready by running the function in the change rows tab. But if you haven't done that for whatever reason, because it didn't fit your data, you can use power rename again, and you can easily add for every plane the end of plane1.tif, plane2.tif in the name. Let's have a look at our demo file to see how it looks like. Right? You see this plane1.tif. This is how it should look like. Alright, now that we have added our measurements, we need to go to the second option, which is the find edges. This is useful mostly for the cellular signal, not the nuclear. However, I only recently made some adaptations for the nuclear signal, and thereby it's actually obligatory for you to run it also for your nuclear signal. Okay, so what find edges is, I also explained on the first video. It's just a Laplacian equation that accentuates the contours of your cells, and by doing that, actually you're getting a higher standard deviation when you're measuring the entirety of your cell. And the higher the standard deviation is, it actually probably means the more crisp is your object compared to the background, because its contours have a lot of intense high values that create a higher variance. Okay, now that we have it, what has happened is that a new folder has been created. We have only one plane again and only one image, so that's why you only see one subfolder here and one file here. If you go inside, you'll just find the same kind of measurements, and what we're interested in this case is the standard deviation. And this is not the same standard deviation that we acquired before, because these images have been pre-processed in a sense with this find edges method, and only after we've taken the standard deviation. Okay, now we've taken it, we've created a file, so what? You need to add the standard deviation to the respective file each time. And now that I've done it, if I navigate to the main measurements folder, you will see that we have a new column added, and this column is the find edges standard deviation. Cool. Moving forward, we need to add our measurements to Arescon again, because our main measurements have now been enriched by the standard deviation column, be opened by Python anyway, so there are later stages that we store our data again. The background mean is what I described before, right? So have a look at these two images. I hope it's big enough at your screen to discern. So imagine that you have an observation, Roy, and what you want to take is that you want to measure its surrounding background mean. However, when you're doing so, you don't want to include signal coming from the row itself, and you don't want to include signal coming from other observations. So imagine that you're measuring, let's say, a region that's surrounding your row. You want to exclude these parts. And this is what's actually taking place here.
With a slight difference, when I did this depiction here, I was using a radius. Now I'm using bounding boxes. So bounding box, right, is just the box that surrounds your object, and I'm just multiplying that bounding box by a factor that you decide, because you might want to take into account larger or smaller surrounding backgrounds when you want to get the estimation for your cells. I'm usually using something like 3, okay, which is quite decent actually to get a surrounding background. Now, auto hotkey, which I mentioned before. What the auto hotkey is about simulating an enderpress in that case. And the reason we want that is that sometimes there is a warning occurring when we're running this step in Fiji, and we just want to skip it. And because we're already running PyAutoGUI, trying to use it at the same time for something else, well, didn't work well for me. I'm quite sure there are some people out there who know how to do it, but I didn't. So that was a super crafted way, let's say, of me, uh, yeah, circumventing the problem. Okay, so we need to locate first the file that simulates the enderpress, and this is the auto ender file. And then you need to go to the program file, so the hotkey UX, and select the UX file. Now you've designated both of them, and if needed, what Fidge is going to do is that it's going to call Arescon, and it's called to communicate anyway with auto hotkey, and it will activate this code. I think we are ready to go. Again, you can see a few instructions about where to find these files there. And let's get the raw mean background measurement. So what it's asking here is that you indicate a folder which will be used in order for Arescon to communicate with Fiji, because during this procedure Fiji will be taking an image, processing it at least to some extent, and then it will be giving it to Python, to Arescon, and Python will do something to this image and will give it back to Fiji. I think it's easier for me to explain what's going on after the function runs. For now, let's just indicate this folder. We can go to the demo, and I like calling it communication. You can also save some sanity checks here if you double click that box there. This is going to double your storage. So unless you have a lot of storage, or just files that are not big, I wouldn't recommend checking on that box. And maybe I can try to explain what's going on now. I should mention here that this is the slowest part of Arescon, most likely, at least. And I think it takes roughly an hour for, I don't know, like a 2 gigabyte image split in 12 planes to get all regions of interest. This also depends, of course, on the number of regions of interest. So you can see that on the panel of image state it says waiting for Arescon to provide bounding boxes of rows. So the idea here is that first you create a convex hull, and convex hull, you can imagine, you can conceptualize it as the polygon that is created when you get the more distant parts of a distribution of regions of interest, and you try to cross the lines from the outer edges of these regions of interest. The reason I'm getting this convex hull is that because imagine that you have some neurons which are lying at the edges of the hemisphere, at the very cortical part, let's say, and you're trying to filter them out based on their surrounding background mean. Now, even if the intensity of the cell itself is very low, or the nucleus itself is very low, the background coming from the real background, which is just the black part of the image, which is outside your tissue, it will artificially make your signal look like it's quite intense, although it isn't. And this is the reason I'm creating the convex hull, because convex hull only gets the tightest region where your neurons or nuclei fit, and by that you can turn any other values of the image to none. And this is what Arescon does here. Actually, it turns values of ROIs and values of background to none. And I will show you now. Let's select yes here, because this is going to make some backups for us. And now it says the metric mean has been passed as a new column. Alright, I think this is a good time to show you what's going on in the background. And hopefully enough, yeah, I have it here, which is like a different data set. And I will try to open an image to show it to you. Alright, I have also overlaid the regions of interest here with their bounding boxes, so you know what was actually counted. So let's try to focus. I know it's not easy. Let's try to find something less noisy maybe. So you can see here that this was a particular region of interest, and what happened is that let's see if we can measure actually. No, I don't want that. Okay, now I can. So you can see that if I try to measure this, there's no value here, because we converted all regions of interest to non-values. And remember, one of the reasons that we wanted is that when you're taking the surrounding background, you don't want your region of interest to also have values, and when you have adjacent cells, you don't want this to impact the surrounding background of this. And this is the convex hull that I told you about, right? You have some marginal, in a sense, cells. Like, imagine that you have a bigger bounding box, you would start capturing the black here, and when you want to filter out cells based on how much does their mean intensity differ with their background intensity, having this is not good. So this is all turned to non-values as well, as you can see here. So that's the idea. Okay, now that we've acquired this data, we need to go back again. We need to go back and add it to Arescon, because as I told you, nothing gets added to Arescon unless you do so. However, there is an autopilot tab that we'll see about later. So background mean is taken already, and now if I navigate again to my main measurements, what you're going to see is that there's a new column created. Actually, two columns. One is the surrounding mean, and this is just the difference between the mean of the particular ROI and its background. I'm not really using that one, but maybe it's useful for you. But I'm using this, and what I'll be doing at the next stage is that I will be checking these values and comparing with these values, and I will be seeing how much larger is this to this by a percentage, and I can place my own filters like that. Something else that is important, although I'm not sure if we'll be able to find it here, some ROIs are actually outside the convex hull. And these are ROIs that are most likely false positives. So these ROIs, normally they have a non-value in the surrounding mean, because really nothing that's around them has any value. But in these cases, we turn this value to 0.00000. This is 5 decimals, and then 1. And this is an artificial value that you might encounter sometimes here. And if you do so, you should know that these are ROIs that are outside. the convex hull or regions that have been haven't survived the threshold. And in this case, what you should do is that you should remove them, and we'll see how very shortly. Now, before I jump to the next step, I should say that this step that we run includes a stage of thresholding. And this stage of thresholding works well for my images, but it might not work well for your images. It should, but it's not guaranteed. Okay? This is about thresholding in ImageJ. What you want to have is a threshold method that creates particles, like the analyze particles option of ImageJ. And you need to apply a threshold method that can detect your hemisphere or your brain if you're using whole slice. As long as you have a method that detects your hemisphere, it doesn't matter if it also detects other objects, because the macro that actually run here in Fiji will only keep the larger object, and the larger object should be your slice. It should be your hemisphere or your brain. So if for whatever reason this doesn't work for you, you can change it from the code, and for that, of course, you shouldn't run the executable version that I run, because imagine that you're going back that folder and you're making some additions to the modules. If you run the executable version in this folder, this is an already compiled code, so it's a screenshot, let's say, right? It's time frozen, so it's not going to work for you. So you need to either recompile or run from Python. So let me show you which part of the code you should change in case it doesn't work for you. Here. So this is inside the run min measurements py module, and this is something that you can simulate in your macro of your Fiji. So just know you might have to change something there. Alright, let's move on now. Let's close Fiji again. Opening Arescon, and the time for the first filters. I'm actually really happy about this tab here, because what you can do is that you can filter rows in or out based on the measurements that you have acquired. And here you can see an example. You can say that I want the mean of my ROI to be at least 30% higher than its surrounding mean. And you can even have mixed conditions there. Like I want my area to be higher than 100, and circumference, I think it's not circularity, whatever, to be higher than 0.9. Okay, and in cases that you have mixed conditions, both of them should be met in order to get a true condition. And only rows that have all their conditions true will pass the filters. All the other ones will be filtered out. The good thing is that you can trace what happens to these that are filtered out, and I've tried to make my whole pipeline like this to have a lot of sanity tests and a lot of accessibility to what's going on in order to be able to trace changes. So here you can say I can do something similar, right? I can say that I want my mean to be higher than the surrounding mean times 1 point, I don't know, 2. And if you don't remember the names of your metrics, you can open the first metric of your first file, and you can see what kind of measurements you've taken and you can use for the filters. So imagine that we have more conditions, as I said, right? You need to have true in all of these conditions, and you can have inside the parentheses like condition A and condition B and condition C if you want to make it more specific, and only if all three of them are true, then the whole thing here in these parentheses will be true. And if you have at least one false, then it will be filtered out. I can run my measurements, but there's no real point at this time. I can show you how they look like from a previous test. So you indicate a folder, and that folder will have your filtered files. It has to be empty, and it creates these two folders, the accepted and the rejected. And in the rejected ones, you can find in subdirectories all the conditions that you've passed. The names will be slightly changed because unfortunately you cannot put some symbols in the names in files in Windows, but you will get the idea. And this way you can overimpose again the zip files that have been filtered out, and you can see if it worked good for your case or not. And then you have the accepted ones. And what is important when you're running that option, you need to select yes for the pickle files, for the creation of the pickle file. And these pickle files will contain all your information about the filtered in and filtered out rows. So keep this accepted rejected rows pickle file, store it quite well because you're gonna need it. After you do the 2D filters, you can move to the Z filters. And this might seem complicated, and to be frank, it is complicated. However, you will most likely stick to the defaults. So, there's nothing to bother. However, if you encounter any issues, you can resolve them. How? Let's see. I'm going to the whole dataset again. Do I have my data here? No, but I have them in the demo version actually. And I have this decision trees folder, which you can also find inside the extra info you might need, and then decision trees in the Arescon file. And I want to show you what's going on here. So there are 3 different colors which correspond to these 3 different columns here. And I make a crude classification of rows as engulfers, which is the rows that really engulf other rows. The rows that overlap with only another row, but they're not really engulfing it, and the rows that overlap with many rows, but they're not really engulfing them entirely, let's say. So what you can see here is the decision tree that is running in the background when it filters rows in and out. Again, to remind you what this is about, this is Z filtering. So this is about keeping the most crisp version of every row that we have across the Z planes and tossing the rest. So I've numbered on this particular file of the nuclear decision tree the nodes. I was lazy enough not to do it for the cellular. I have to do it at some point, but it's the same numbering, so you can trace issues by looking at this file as well. Now the decision trees differ a bit, but there are no big differences, and you should be fine. So I've numbered the nodes, and here you can see also the conditions. When you have a blue arrow, it means true condition. When you have a red arrow, it means it is a false condition. And you can see when something gets included or excluded. Now this of course requires a bit of familiarity with some terms like intersection over union, things like that. But if you focus on one particular node at a time, you will see that it's not so tragic to understand what it means. If you can't understand what it means, of course, you can message me and I can help you with that. But most likely is that you won't have to change any of these values. At the same time, when these values are created, so I actually have it here. This is another file with CIFO signal. And this is the folder that is created once you run the Z filtering. Okay, you apply your filters, you leave the defaults, you don't have to write anything. If you don't write anything, you will just take the defaults. If you want to change something, you go to the particular entry field and you change it, of course. And then when you look inside this folder, for every final, let's say, in a sense, set of regions of interest that you have, you can also have you will also have these Excel files, and these Excel files tell you for each particular row where was it, if it was included or not, and which node led to this decision, and if there are more conditions on this node than whether it was true or false. Okay, And this way, if you see anything that's peculiar in your final filtering, you can first get into these Excel files, find the number of your ROI, and after you find the number of your ROI, you see the particular node that it was included or excluded. And as I showed you, the nodes are numbered. And if you want to change the value on that node to try filtering again, then you can see with the Latin number which value it is. Imagine that something, actually 6 is not a proper one because it doesn't decide. Imagine that you have node number 7 and you want to, something went wrong there and you say, okay, let's make some changes. Then you see that it's about this particular value, okay, which is Latin 6, and you can find Latin 6 here and you can change it. This is the idea. I understand that it's not very easy to understand, but it's not as complicated as it seems, trust me, and you can always ask me if you want. Okay, so this is where the previous demonstration ended, and I can show you again how some regions of interest, sets of regions of interest look like in the end. Let's open this file, which is the file that I initially showed you, and let's see how the filtering looks like now. I'm going to show all. I have to say I haven't done much filtering here on the X and Y axis, so it can be improved. And it's quite interesting to see the Z axis as well. So if I try to come here, I'm also going to install a plugin that will make my life much easier in terms of visualizing. I have this show all file that allows me to turn on and off the regions of interest. So these are all the final detected regions of interest in your file, and let's use our real ROI names as labels, and let's have a look. So we're coming here, and now I'm imposing the regions of interest, and you can see that it works quite clearly. And here's the thing, that if you don't filter across the Z-plane, you'll have one observation, one ROI for that, but you might also have one ROI for that, pretty much. You might even get one ROI for that, so it will be very messy, right? Because instead of one ROI, you have 4 or even 5 or even more. But you can see that the Z filtering worked fine. Let's go to a bit more demanding, let's say, parts, and see how well it works. And as far as I see, it works quite well. And here you can see that here, for instance, you can't see clearly the neuron. Let's see if you can, yeah. So it actually captured it because it could find it on another plane. Yeah, actually I think it's pretty good. Just to convince you, I'm going across a few more densely populated regions. Someone would certainly go to hippocampus here and see how well it works. And I think it's actually quite amazing. And this is not on me, this is on the people who created Cellpose. I'm just doing the filtering, I hope, properly here. That's of course based on your subjective criteria as well. But I believe that a good job is done in terms of the filtering. Now there might be some situations, actually this seems to work okay. There might be some situations that there is miscalculation in the number of the cells that lie in the region. But you get the point, right? You look across all the planes and you overimpose your final regions of interest in one image, and you can even do a Z-max projection in the end if you want to overimpose your regions of interest to have a more easy look at it. Okay, I think I've looked enough at regions of interest. Here there might be a mistake. There seems to be an engulfer, as I call this, is actually one too many overlaps, and this particular one should be out. What you see here is the echo of the other two images, more likely. So the signal just spreads when you move across many planes, and this is the reason it happens. But for that, again, we could go back to the filtering of the default and change it if it's many occasions like that. Here we have another occasion like this, right? Which one is it? I think it's the 7. Let's see. Yeah. As you can see, this is the mere outcome of echoing signal from two different cells, and by changing the defaults, that should be taken out. And if you see some small regions of interest like here, this is because I haven't used filters for area in the XY filtering, but you will do it and you will have no problems like this. Alright, so that's how you get your files, and if you're using cellular signal as well, like CFOS, EGR2, ARC, whatever, you will also get quite good filtering. I hope so, at least. You should. And the filtering there, when it comes to the crispiness measurement that I mentioned also to the decision trees, is based in the mean value for the nuclei and the fine edges for the cells. You can see at the end of the names, I also mentioned in the first video, this does, and this indicates where Arescon believes that the most clear version of a neuron is based on these dashes here. Cool. Now let's jump to the new stuff. Overlaps. Well, you might study colocalization, you might study reactivation rate, a lot of different things, and you need to get some overlaps. First of all, you need to select the folder with your large regions of interest, and by large, it's probably cytoplasmic, cellular in a sense, and the small rows, by the small rows I mean the nuclei, because I want to see to what extent do the nuclei lie inside the cytoplasm. Names have to be the same between these two data sets. So here we have a folder with final regions of interest after they've been filtered for the cytoplasm, and here I have a folder for the nuclei. So I've run this whole procedure twice, one for the one channel and another for the other channel, and now I want to get the overlaps. You need to select the large folder first, and inside the colocalization folder. Now I'm going to select small folder, easy as that. Just indicate a save folder. Let's do it in here. Let's call it overlaps. And then you need to indicate what kind of overlap you want. And I'm using, taking from mathematics, but I guess you get the idea. I'm using the intersection symbol to indicate that I want to see the intersection region between the nucleus and the large row, which is the cell, and how much of the nucleus's body is inside the cell's body. Okay, so this is what this means. You can also go for intersection over union, but I haven't tested that thoroughly, so I'm not even sure if it will work okay, but you're encouraged to do so and let me know, of course. And what percentage? I want, let's say, at least 80% of my cells to be inside of my nuclei, to be inside the cell. And because in my dataset I had some bleed through, there's also an option that it will take out observations that really look like the cells, so nuclei that really look like the cells, by getting the intersection over union. Again, this is very particular, very specific, probably not for you. I hope you have better microscopes than the one I used. Lastly, we have to correct for a larger nucleus, maybe not. So the idea here is that sometimes, imagine that you have a small circular cell, and you have a nucleus that it has so much signal that it makes it look much larger, and it might actually be sort of larger than the cell itself. It's a very particular case as well, but you can also correct for that. And you can say that in these cases where the nucleus is overlapping with the cell, but the nucleus is actually larger, well, be a bit more lenient, let's say, be a bit more generous and liberal in terms of estimating the overlap. And I don't know, let's do 75. Okay, if everything has been put correctly, you should get your results. Let's see. Okay, let's have a look. Oh, here, the overlaps, yeah. I was dumbfounded for a moment. So here what we have is the final regions of interest. If I'm not mistaken, they're based on the not the intersection per se, but on the cell. But I really have to check about it. I don't remember by heart. But you have the final regions of interest in zip format. So all your final files that you want to have, right? If you're looking for reactivation rate, for instance. And you can also see, again, like the logging file, and you can see the threshold that you used if you wanted to correct, and what threshold you put for the correction. And you can see the percentage of the overlap and which neuron they were overlapping with. A bit more detailed here, but still useful if you want to go through some things. And having run the overlaps, the other thing that you can do is that you can imagine that you have drawn already some regions manually. You can either do it manually, or there are some really nice pipelines out there, which unfortunately I haven't experimented much on, like Quint, for instance. Quint actually does the whole thing, not just the atlas registration, but also cell detection. But you can isolate this part of Quint and incorporate it to Arescon, for instance, and eventually you want Fiji designated regions, that is ROI files, that you can use in order to map your detected regions of interest to these designated regions of interest. Let's try to do that now, very fast. Alright, so I've opened my demo image again, and I'm going to adjust the brightness, and I will just pick two regions. You can do as many as you want, of course, right? I'll be very, very rough and crude here. I don't mind precision at this point. Okay, and I don't know, let's just take this. And let's name this region A, or just A, and let's name this B. Alright, and you can have as many as you want, of course. So I'm going to save both of them. Let's save them on desktop. I'll have to change the name though. And let's see what name we have to insert here. Okay. Alright, I'll have to rename it, and I will rename it based on the file name of the final images, which does not include the plain number, it's just the plain part in the name. So, oops, yeah, of course, if I do it here, I'll have a problem because I will override the previous one. So let's create two separate folders. Let's name one subregions, and in here I put all the ROI sets for every particular image that I have. And let's call the other one, I don't know, ROIs maybe. Alright, so we select the folder with the zip ROIs, and this is that, and the folder with the subregions, which is this, a save folder as always. I'll name it save. Minimum intersection of a particular roid to the subregion. This is because when you're designating subregions, you might actually slice some rows, and you need to decide what percentage of row should be there compared to its body in order to include it. Minimum is 51, and you can go higher than that if you want. I'll go for 70. And yes, I want to generate some rows for sanity checks afterwards. So I'm clicking map to region, and that went fast, but let's see if it worked. So let's go to the save folder, and we have something, right? Let's see. Yeah. So here you have your final Excel file. This is what you want for your analysis, right? It's just the listed regions and the number of observations they have, and you can do it for one channel, for the other channel. You can do it for the overlaps by placing there the zip folder containing the overlaps, and by that you can know exactly how many neurons you have in every region. And if you want to also run some sanity tests, let's open the demo image again. And let's navigate to the save folder, and let's place the regions of interest for the A and the regions of interest for the B. And where's my right manager? Here. So you can see both the designated region and all the regions of interest that are inside there. And these are some occasions that I mentioned before, right? So this is less than 51, so it's out. Likewise for this one. Alright, we're almost done. Autopilot. What is autopilot? Autopilot is a way to run all the filtering automatically, right? Because disadvantage of Arescon, as you see it here, of course, is that you need to wait until a tab function related to a tab is done and then move to the next one. However, with autopilot, you don't have to do that, and you can select if you want to finish at the 2D overlaps or at the 2D filters, or if you also want to perform the Z filtering part. For that, you need to indicate again all the options that we indicated here, right? So again, all these buttons here, all these entries, the images folder, the rows folder, save folder, the image.jpg file, the seconds. You don't click generate measurements though. You leave the add metrics. This is going to be automatically. The row enlargement factor, the files again in here. Your filter should be written here. You won't press apply, but you need to put your filters. Any potential changes that you might make for the Z filtering, and that's up to here. And you can run the autopilot, and by running it, if everything goes well, which you should get the whole filtering part done. And one last part that I should mention really fast is that for images that you have weak signal and you still want to maintain that signal, you can pre-process your images. I have a script inside the Arescon folder for that. Let's try to find it. This is in Fiji macros, and this is the pre-process for macro. You can ask me if you don't know how to run Fiji macro code, and the idea is that you can solve it. You can boost the signal of some regions of interest, because some regions don't have strong signal anyway. So maybe in these regions you want to solve it, the weak signal. So you can boost the signal. Here I'm having regions of interest from a pre-processed and non-pre-processed image. the image itself is the same. Okay, so it's just the regions of interest that I'm showing here that is different. Um, and what you can do is eventually you can combine this signal, and this is actually quite useful, and especially if you're trying to uh, make the filtering based on the mean background that I showed you before. You might lose some observations unless you have this merge-Roy inferences option inserted, because imagine that CellPost hasn't detected some adjacent cells to your cell. If that's the case, then when you're calculating the surrounding mean background, unfortunately you will take into account the values of the dim cell that hasn't been captured by CellPost. So by boosting the signal, you also ensure that even these cases of non-focused dim cells will be there, and by having them there, you're making a better filtering for the rest of your nuclei. Cool. I think this is that. I know it's quite a few things, but yeah, I hope it will work well for you. And as always, feel free to ask any questions you might have. Thank you very much.